Hello, everybody, and welcome to the Smoko podcast. My name is Alexis Armstrong, your host. Nice to meet you. The Smoko podcast is the place to celebrate and highlight women, trans women, and non binary folk working within STEM and trade occupations. So please tune in, take a break, and join us. We are on Smoko. And today we're extremely lucky to be joined by Mia-May Grabovac, who is a student at the University of Manitoba. She studies marine biology, which I know there's probably so many people out there listening that'll be like, oh my God, that was my dream to become a marine biologist. She is becoming a marine biologist. She's in her advanced years at U of M, and she's thinking of going to graduate school and graduate studies. So we're going to talk a little bit about that. But we're also going to talk about her really cool work that she's done already as a research assistant and as a student aboard the Canadian Beaufort Sea Marine Ecosystem Assessment. And so we're going to talk a little bit about what that work was, her study of zooplankton aboard a research vessel, and marine biology in general. So, Mia, thank you so much for coming on the show.
Yeah, thank you so much for having me. I'm really excited to talk about my work as a student and getting into marine biology.
Oh, I'm so excited to learn more. As like someone, a fellow who started in marine biology, that was my background as well in biology and geology. I'm so excited because I love I love talking about the oceans and talking about the critters of the ocean. So
Yeah.
really excited to have this conversation. And maybe if that could be where we start,
Yeah.
is how did you become interested in science and then specifically marine biology? How did you discover this field? And then how did you go from discovery to be like, okay, you know what? Like I'm going to become a marine biologist. This is my path.
Yeah, for sure. I think I've always had interest, especially as a kid, in biology. And my actual goal before I even kind of started to think about it was I wanted to work with sharks. I loved sharks. I still love sharks, but I kind of always expressed an interest in that. And I think my kind of interest and passion for biology really sparked towards the end, like the beginning and end of high school. You kind of go into biology more specifically. I know grade 12, we focused a lot on like animal
Mhm.
biology and genetics and just evolution. So I kind of took that and then I applied direct entry into science at the University of Manitoba. And I was lucky to get accepted right away. And when you kind of go into the University of Manitoba, I think they're changing it now, but they have a lot of different concentrations that you can go into in biology. So they have evolution and biodiversity, which is the concentration I was in. They have cell biology, integrated biology. And I knew that I wanted to explore my options and take as many courses as I could. And with that, I kind of started with the basic biology courses, and then I found a real interest in inverts. So there was a
Okay.
class called it's like Introduction to Invertebrates, and a lot of that is focused on kind of marine life. So we look at sponges and the corals, the jellyfish, all that, and I kind of fell in love with that aspect of it. And there was another course, it was marine biodiversity.
Mm.
We focused a lot on kind of the history of marine conservation and where it is today. We did a lot of debates on a bunch of topics such as like anthropogenic issues, climate change, how that's affecting marine systems. And I kind of fell in love with that. So it started to kind of like curve into, oh, I want to do biology. I want to focus on conservation for the oceans. I know coral reefs was a huge one with the bleaching that's going on and how that's affecting a lot of the environments for the animals that live in those ecosystems. But kind of gearing towards my third year, and a lot of my degree was done over COVID, so it was an
Mm.
online online portion
Very
of the
different.
degree. Very
Yeah.
different. I kind of lacked that lab experience
Mm.
that you kind of are supposed to gain during your university time. So we did still look at anatomy of animals and things like that, but it's not the same as going in and
No.
doing your hands-on work.
Yeah.
So kind of end of my second year going into my third year, I felt very lost at what was I going to do. I didn't know
Mm.
how I was going to get a job. And it was actually one of my lab partners in one of my classes. She recommended that I joined the University of Manitoba as a science co-op program.
Okay.
And in co-op, you do three kind of school terms. Instead of doing classes, you actually look for a job. So they teach you kind of valuable interview skills, how to create a resume, how to create a CV. They go through practice interviews with you. Then you could apply for a bunch of different positions. And I had zero experience, obviously, outside of just kind of retail jobs and those types of odds and ends. So I applied for everything that I
Mm.
could. My first co-op job, an introduction to science, actually, I worked at the University of Manitoba.
Oh, very cool.
I worked kind of involved in a goose management plan. So the campus geese are very mean. Um, so I kind of looked into our policies around management for the geese and how we can kind of coexist with them because
Mm.
they're not going anywhere. They're huge population over here. So that was kind of an intro. And then, luckily for my second and third work term, I got a job at the Department of Fisheries and Oceans Canada
Mm.
under the Freshwater Institute, which is also on the University of Manitoba campus. So, kept it kind of on campus for me. But I worked in the Arctic Aquatic Research Division under an ecosystem stressors group. And
Cool.
a part of this group was the Canadian Beaufort Sea Marine Ecosystem Assessment Program. So it's a program that's been running since about 2017. And they kind of go into the Canadian Beaufort Sea, which is up in the Arctic, and they run annual monitoring of the ecosystem in there. So they look at oceanographic conditions such as salinity, temperature, chlorophyll A. They do fish,
They do
zooplankton,
everything. It's a
everything.
complete ecosystem
Yeah.
study. It's a full top to bottom ecosystem study.
Yeah, exactly. So I started as a laboratory technician in my co op work term. And through there, I kind of gained real passion for Arctic ecology and marine Arctic
Mhm.
ecology. I never expected that I'd be, um,
Went from sharks to the Arctic. It was a
yeah,
very complete different change. Yeah.
exactly. Going from such like a big fish and a big animal that I wanted to do to like the smallest possible.
Zooplankton.
Yeah,
Yeah, the complete other side of the pyramid of the
exactly.
Well, I love that. Like, I love that story. And thank you for going into the full aspect of like what part of sciences you were the most interested in and where you
Yeah.
fell in love with it. Like, what courses kind of got you hooked and more hooked the deeper you went into marine biology. I love that it's a very clear shot. And I feel like that's not something really normal or seen
Yeah.
within these fields. A lot of people sometimes don't know that mechanical engineering or geology exists. But I love that from a young age, you were like, yep, I am in love with sharks and I'm in love with marine biology. And so that's the way that I'm going to go. And I especially love it being from Manitoba, being from a
Yeah.
landlocked, a landlocked
That
province.
yeah, I kind of didn't I did think about moving for my undergraduate.
Mm,
There's a lot of good programs on the East Coast.
mm.
Memorial University stands out in Newfoundland. There's a university in New Brunswick that's very good. There's two campuses, but I can't remember specifically which one.
I
But it's
actually
just
went to school out east for marine
oh,
biology. I went
really?
to Acadia. Yes. I was like,
Oh,
yeah, there
yes,
was great
Acadia
programs
too. Yeah.
for maritime. Yep.
So I definitely thought about that, but I was like, I'm home here. I grew up in
Hmm.
Manitoba. You could do a biology degree. I did kind of have plans to go into a master's. I guess we'll talk about that later. But
Yeah.
I was like, maybe I'll just go out later. And I had no idea that we had a fisheries and oceans
Mm,
here, and I had no idea that
mhm,
a part of it was in the Arctic, which I find really cool. That
mhm.
a little like landlocked Manitoba has a
Yes,
really big portion of Arctic research in Canada. So
I think I think it's fantastic, and I love that you brought that up because I think it's worth mentioning that you don't have to be coastal to be involved with
yeah,
this science or in love with this science. And I worked for years for the state of Texas down in College Station, and that has one of the best oceanographic research programs in the entire world. Again, same thing, it's landlocked.
yeah,
Like you wouldn't expect small landlocked Texas, especially that part of Texas, because that part is landlocked,
hmm
being this kind of hub of oceanography. And then the same thing with Manitoba, that you guys are actually a big hub of Arctic ecosystems and
yeah,
understanding the Arctic. So I love that you had that passion and then you went into marine biology. I love that it also turned into marine invertebrate. One of the coolest courses I ever took was marine invertebrate zoology.
it's
And it was partially because of the lab. I had a crazy lab professor that we would do the dissection and then we would eat them. Like he'd bring in scallops, like just like fresh scallops from the grocery store,
that
and
is
then be like,
crazy.
dissect them. And then like you had like 10 minutes. Like he was like, okay, they need to go in butter and wine and garlic. So like, come on, hurry up. And it would be like mussels, scallops, and lobster was like our main dissections. And it was the best course I've ever taken. It was
That's
literally, it made me a marine
crazy.
biologist. Yes. Could you imagine that you actually eat your dissections? I called my dad, who was also started as a biologist, and he was like, "Excuse you, what?" And I was like, "No, like it was actually like they were live specimens. This is how we ended up." But
What a way to
um,
learn. That would be so
yeah.
cool.
Yeah, honestly, it was I'm so sorry that that wasn't your experience being in
No.
COVID.
I wish. Dang.
Oh my goodness. Yeah. I'm so sorry that that wasn't, but I love that you ended up in the Arctic and you ended up working for freshwater and for Manitoba. I love that you did a hilarious geese study on campus.
Yeah. That
As
was
a ch-
interesting for sure.
Oh my goodness. Was it just wrangling geese? Because I feel like the practicality of biology is so funny compared to the theory.
Actually, luckily it wasn't. I think
Okay.
that would have been very scary. But I did do a just like a very small mock experiment study where we had some previous data from 2019 and 2018 of where nests were located on
Mm,
campus. So in 2022, when my co-op work term was, I went out and kind of counted where nests are approximately, and I kind of did a comparison just to see if any of the nests had kind of migrated, maybe kind of moved down further south. And it was actually very interesting because we're located right on the Red River. So it's kind
mm,
of very close. Like there's a big kind of, it's a hub more for like for the geese and for ducks and
hmm.
for other
It's a
wildlife.
big estuary.
Yeah.
Yeah.
Just right out of camp, right outside of campus. So it was interesting to see that when the campus was unoccupied, the geese actually moved more into campus because there were no people. It was a bit more safe for the from the
Oh.
predators. They
Hmm.
used the buildings as protection essentially. So it was interesting kind of where they had moved, especially
Mm.
when we were essentially
Gone.
not yeah, gone.
Yeah. Yeah. When you guys were doing your labs as best you could
Yeah.
online,
Yeah.
the geese were finding security and safety. That's a really cool study actually on like kind of population and geographic mapping of like
Yeah, exactly.
hotspots of geese. That's fantastic. And I don't know for the next one, Mia, if it's the geese or if it's anything that you've learned in your co-op of working up north and working in the Arctic, but throughout your studies in marine biology, I know you started as big species and you ended up as phytoplankton, like you ended up as the smallest primary producers. But has there been anything along that journey within the science that you've learned that has maybe changed fundamentally how you view the world, or has been something that really melted your mind the first time you heard it that you were like, oh my goodness, I have no idea that that was a thing?
I think definitely working in Arctic
Mm,
ecosystems, I didn't realize how the Arctic, like especially the poles, Arctic and even Antarctica, how
mhm,
affected they are by climate change. And it was actually very recently that I took a class in, it was like an introductory environmental science course. And I thought it would be really cool and important to take. And I kind of determined how much a single individual has effect on these ecosystems, and not just in your immediate area. But for example, a big
mhm,
thing or a big issue is mercury in the Arctic. And mercury travels really long distances and for a very long time in our atmosphere.
mhm.
So even industrial areas like big hub, like city hubs, mercury production from those hubs can travel all the way to the Arctic. So it kind of blew my mind that we're so far, but we're having
Mhm.
immediate effects. And kind of crazy to me. I was like, oh, made me really reflect on what I do every day in my own life that we kind of
Mm.
have to realize for ourselves. So,
No, thank you for bringing that up. And I think that's such a good point. It's a sad point and it's a scary point. Like it has a lot of consequence to it. But I think that I was under the same impression before I worked polar research and before I was in a polar region is I thought that they were still these really untouched
yeah.
wild places that maybe didn't have the same effect of climate change or global warming as the rest of the country or the rest of the planet. And I will always remember when we took on the Geordie's resolution, we were able to take a deep sea sample and a deep water sample of Antarctic bottom water
Mhm.
by the end of the drill bit at 3,000 8,000 meters. And so it was deep, deep water. And it's also it's the oldest part of our marine thermosaline circulation, right? So it's really, really old water. It's very deep water. It has a slow recharge rate. And within that water, we actually found microplastic. And it was something that will always stay with me, that I was like, how dare we that we found microplastic
Yeah.
in that environment and in a system that is so slow on the recharge and is so slow on the circulation. And the fact that we still had like a anthropogenic impact and visible data that we could see under the microscope and be
Yeah.
like, everything that we see right now is covered in this thin film of microplastic.
Yeah.
It will always stay with me.
That's crazy. Yeah.
Yeah. But I think it's very similar to mercury. Like it's similar to be like, okay, well, within these areas, you're seeing an industrial spike of the water chemistry with mercury or the water chemistry with iron or whatever you're looking
Yeah.
at. You're looking at an industrial byproduct. You're still seeing it within these marine ecosystems.
Exactly. And it's such a big kind of cascade down this chain of, well, it's in the atmosphere, then it gets into the water, then it gets into the animals, then the
Hmm.
communities up there who rely on those animals, like seals is a really big one, are affected by mercury poisoning. And you don't even realize it until you start to see that kind of long term. So,
Hmm.
yeah, it's crazy to think that your everyday life affects like something
Animals.
so far from you.
Yeah. Yeah, completely. I remember one of my marine biology professors, he did a paper and it was the same marine biology professor that let us eat our experiments. So he was a wild, wild man. But his paper was called It's a Merry-Go-Round, Stupid. And it was looking at the migration patterns of different populations of salmon and the effect of cesium from Chernobyl and how that cloud had come over the North Atlantic and how you could see different levels of radiation and cesium within salmon based on where they were within their migration pattern. So you could then tag salmon using cesium, using like you could use them an element that's not natural to that system, and you could use that signature to try to figure out circulation. It
Wow.
was an insane, insane paper, but it was all based on human impact and on Chernobyl and on that cloud of Chernobyl and then how it affected the ecosystem. It was wild,
Yeah.
absolutely wild.
Yeah.
And maybe if we could talk a little bit about the sensitivity, because I know that's a big part about why you guys are up in the Arctic,
Yeah.
because it is kind of a canary in the coal mine. But with the Canadian Beaufort Sea Marine Ecosystem Assessment, I know that you talked a little bit about how you guys do everything from the top to the bottom. You're just doing a full ecosystem assessment and survey. Could you kind of describe the projects, its initiatives and mission, and then maybe briefly the experience of what it actually meant to go out and to do that research and to live on that research vessel?
Yeah, for sure. So we just call it CBSB.
Okay.
I know it's like the title is really long, but CBSB is totally fine. So it's just an annual monitoring program focusing on relationships between the environment and the animals and the response of each ecosystem within the environment. So everything is very sensitive to change. Zooplankton, for example, is a pretty big one. And these changes can have long-term or drastic effects on the other parts of the ecosystem, from the phytoplankton to the bigger individuals such as the bowhead whales. And
Mhm,
these effects, they don't happen overnight, right?
mm,
So this is why kind of annual monitoring and assessing the oceanographic conditions of the Canadian Beaufort Sea in relation to these other individuals is very important. So we do a bunch of different assessments, as I've kind of mentioned. So we look at water chemistry, chlorophyll A concentrations, which can tell us the abundance of where the phytoplankton are, which can kind of give us an idea of zooplankton abundances. We do benthic invertebrates. We do fish. Arctic cod is a really big one that's up
mm,
there that we see a lot of the times. Some objectives that we kind of look at is what are some ocean drivers of species distribution? So,
mm,
how does salinity affect distribution, chlorophyll A concentrations, temperature, especially with kind of increasing climate change, is a really big
mhm,
focus that we look at. We also look at the stressors that are present. So, ocean acidification is a very good
Yeah.
example, and we can look at that through the kind of shells of the benthic inverts. So,
Hmm.
calcification can be extremely affected by how much acidification we see. So, we kind of look at those and then how they're affecting the rest of the environment. A big one is we focus a lot on communication with the indigenous communities that live up
Hmm.
there. So they have a lot of requests for monitoring around the Inuvialuit sediment region is where we kind of sample around. So we often comply with the requests that they want to see, and we make sure we're always kind of in close communication with them, making sure that just kind
Hmm.
of
Yeah.
big communication around there. So it's a very big kind of collaborative program. And
Hmm.
we build off each year just to compare what the conditions were last year versus this year. We can use that to kind of understand what's going on if a big concern is climate change, for example. I know for an example, on my when I went last summer in 2023, we saw a very open free ice year, which was very drastic
Oh,
from last year
yeah.
where they had a couple of sections that were really, really icy. And I remember I was told that they almost kind of got stuck in the ice, which was really scary. But this year we saw little to no ice. So even just the sea ice duration, how long it is, that can really imply for
Hmm.
phytoplankton bloom, which can accelerate when zooplankton
Hmm.
kind of are abundant, affects how it's just the whole kind of big cascade
Everything's
in the food web.
interconnected. Yeah.
Exactly. So that's why the annual, like the monitoring of everything is so important because it's not just one factor that affects everything, it's a multitude that we need to kind of keep watch. Yeah, and then kind of going back living on the boat and kind of what that looked like, I had fun. Super it was super interesting. I know that fieldwork is a pretty big aspect of biology and marine biology especially, so I was very lucky to get the opportunity to go onto the boat with all of these amazing researchers and scientists, especially as a student.
Hmm.
So I really gained a lot of valuable experience seeing what it's like to live on a boat for a whole I lived on it for 5 weeks. So
Oh, wow.
we slept on it, we
Yeah.
ate, we had a very lovely chef. She made us very nice meals. A big thing that I kind of learned quickly was maintain positive attitude because
Yes.
you're stuck with everybody. So but
Yeah.
it, it was really interesting to hear all the stories that people had to share about their time entering the science field, and especially coming from working in the lab for so long
Mhm.
for as a co-op student. It was interesting to see the collection and do the sample collection itself, and it came full circle for me to be like, oh, this is how we
Mhm.
collect it. I process it in the lab and then we send it off for analysis. So it was very like that merry-go-round almost. It was like,
Yeah.
oh, this is this is like the part that like I didn't that I got to experience, and I was very, very lucky. So
Oh, that's fantastic. No, that's really cool. And it's cool that you got to see the full scale of science. And I think that as a scientist, I think sometimes you honestly forget about it to be like, okay, how big and how compartmentalized it is of getting data collection is just the first step. And then it's a lot of lab work. It's
yeah.
usually back and forth between those two, honestly, continuously with time. And then
Yeah.
the third one is then analysis and then finally publication. But it is this full system. So that's beautiful that you got to see the project on terms of data collection and boots on the ground, and then also tie it back into your co-op work
Yeah.
and the work you had previously done. I love that you loved the experience of living at sea. I think there's honestly nothing like it. I'm obsessed with it.
Yeah.
I always reminded me a little bit of living in a time past or a time
Yes.
gone. Like I felt like it was really a scientific expedition from the 1960s or even earlier. There was this energy to it that it just felt like you were stepping out of time.
Yeah. I felt very almost not disconnected, but it was nice to kind of focus just on work and building connections with your coworkers and having a laugh. We kind of quoted it as like a groundhog day where you kind of like live the same day over and over.
Yes. Yeah.
But it's still so much fun, and it's never a dull moment. Obviously, something can always happen. Communication's a really big one
Mhm.
as well. Like talking about what we're going to do for the day, what's planned. We have to make sure that in the event that something happens, we're prepared for it. What the weather's going to look like. It's a whole big collaboration, and but I love it. I love kind of being in that just moment,
Yeah.
like barely, like, we had a service and stuff on the boat, but you don't have time for that because you're so
No,
busy.
you're working.
Yeah, exactly.
Yeah, it's like it's this beautiful kind of environment that you're like, this is so hard to do in any other space of getting all of these scientists together, working on something bigger than ourselves. You do kind of have to have this authenticity and honesty on boats
Yeah,
because, as you said, like you're living and working with these people. You have to be kind, you have to be
yeah,
upfront, like you have to have good communication. It's a beautiful and very different environment working in a research
exactly.
station. Can I ask one follow up on the actual work itself?
Yeah, for sure.
I think it's so fantastic that we're doing all of these surveys and we're getting everything from top bottom as hopefully people listening can maybe understand of how interconnected it is. If you move one lever in terms of environment, it's going to have consequence and it's either going to be on population number or maybe distribution of that population. It's going to look at diversity and species diversity, but it's also going to change maybe chemistry, change salinity, change temperature. Everything is interconnected. When you're collecting all of this data and you're looking at it annually, what are you comparing it to? Is there a base measure of maybe this is what a healthy Arctic should look like? And if we've strayed this year or this summer, or we have this bloom of algae, or we have this drop in oxygen in the water column, that's obviously unhealthy and unstable for the environment. Do you have a healthy monitor that you could then compare it back to?
The kind of assessments have been going, I think it's more based on just year to year. There
Okay.
are kind of expectations. For example, for a marine system, your salinity should be around 30 of
Hmm.
the
Yeah, you're
PSUs.
Goldilocks.
Yeah,
Yeah.
you're kind of, so you have your baselines, but I think it's more important to focus on the changes
Mm,
rather than referring back to the baseline.
okay.
I'm not 100% sure on this
No, no, no.
kind of
It's
topic, but yeah, I know for sure, like, for zooplankton, like, a lot of the times, there's never really a like, oh, we should expect to see this. We have kind of ideas based on species composition.
hmm
So copepods is a really big, it's 90% of the zooplankton, makes up about 90% of the composition. So we have expectations of we should see mostly copepods when we do our assessments or we take out or look at our collections. But if we don't, then we can kind of be like, it's a bit
okay. What's
weird.
happening?
Yeah.
Yeah.
Or kind of compare it to last year's where it's like, well, we had so many of these and now we have virtually none. Something must be going on for this
Mm.
species. So yeah, I think it's more focused on long term. But
Year to year.
yeah, but we've started, or the program started, it was under a different name. I only know the acronym, it's
That's
BRIA, but
okay.
it started in 2012. So
Okay.
essentially almost 10 years of assessments and data that they compare to. So
No, thank
I
you. Though
think that's
that's
pretty crazy.
it is crazy of 10 years is 10 years is a lot of time,
Yeah.
right? And that's fantastic that it's still an annual thing and the richness of your data is so so important and I think
Yes.
that's probably the best part about this program is just the variety of data collection and the fact that you're looking at it as a complete ecosystem is really beautiful. And I love that that's where you're making your hypothesis is from year to year or general trend as you compare them other than a baseline. But I just wanted to establish maybe how you're looking at if an environment
Okay.
has changed or like what does that actually look like. So that's wonderful that it's
Yeah.
on a year to year comparison.
Yeah.
Fantastic.
Mhm.
And if we could dive into a little bit about the zooplankton,
Yeah,
because
for sure.
those little copepods, those were your focused, and you did the delivery of something called the rapid zooplankton assessment, which I
Yeah.
know no one probably has any idea what an RZA is. So maybe if we could take a little time and explain what that actually looks like. And then what's the process of sampling? Because again, I think if we explain like, okay, we're collecting this zooplankton, people might not understand actually where zooplankton are or like, is it a big net? What does that actually look like when you're doing that task? And then what's the purpose of it? What are you guys hoping to research within that study of RZA?
Yeah, for sure. I'll go kind of into the background
Okay,
first,
please.
if that's okay.
Yeah.
So, zooplankton are kind of key forage species for a lot of the different kind of animals. So, they're an important link in the food web. They take essentially what the phytoplankton makes, the lipids, and they convert
Mhm.
those and they store them in their bodies. And important species such as Arctic cod and bowhead whales, they don't make those lipids, so they need to take them from the zooplankton. Copepods are a really good example. They have a lot of fat storage, so Copepods are often a go-to for these animals to consume, and then they're able to kind of get those high energy lipids from these copepods. Copepods also do what's called a geovertical migration. So
Okay.
during the day, they kind of stay closer to the bottom, and then at night, they migrate up. So this is also beneficial for a very important link between the pelagic ecosystem and the benthic ecosystem. So a
Mm-hmm.
lot of the benthos you can see will kind of eat either dead copepod zooplankton or their kind of fecal pellets.
Detritus.
Detritus, yeah. Marines, we call it marine snow. It's kind of like
Yeah.
all the things that kind of go down, whether that be kind of if they've shed exoskeletons, anything like that. So they are an essential link in the marine Arctic food web.
Mm.
And they are very sensitive to kind of changes in oceanographic conditions. A big one is increasing temperatures in the Arctic. So, an increase in temperature, as we know, can kind of cause sea ice duration to be shorter than it has been in the past couple of years. This can change the timing of the phytoplankton bloom, which changes the timing of when zooplankton are most abundant. And that can also affect foraging species or the planktivorous species such as the bowhead whales and the Arctic cod. So we want to make sure that we do annual abundance assessments for these zooplankton. We also do biomass assessments, community composition, so which species are there, which species are most abundant. So copepods are a big one, specifically Calanus hyperboreus. We see those a lot.
Okay, very cool.
Yeah, but we kind of do a big monitoring thing, and abundance assessments are very important in the sense to monitor for their predators. But when we do abundance assessments, we do a traditional taxonomic method. So we do a zooplankton collection, which I will get into of the more specifics, but we often send this to a lab. It takes a team of experts to do, and it takes quite a bit of time. It takes about a year, even more than a year.
Oh, wow.
Yeah, so
That's a
we
huge amount of time. Okay.
yeah, we just actually got back all the 2022 data. So it takes a long time to identify all these species. So recently there was an ecofoci group, and they developed a rapid zooplankton assessment, which essentially, when you collect the zooplankton, you can take a subsample of the collection, so about 10 milliliters, and you do a quick count of about 100 zooplankton within that under a microscope. And then you can kind of get real time or quick abundance assessments or estimates, which can kind of tell us or give us an idea of where the zooplankton are abundant. Specific kind of groups like the copepods or amphipods is another big one that we look at. And you want to do this also annually to kind of compare the estimates from last year to this year to next year. So as part of my co-op project, I worked on this method to kind of compare it to our taxonomic data. And I did this using already preserved samples. So the specimen were dead. I just kind of did like what we would do on the boat, but just in the lab. So through this, I was able to kind of determine how comparable the abundance results are from the RZA method to our regular methods. In the end, we need both methods. Taxonomic data is still super important for abundance assessments. You get the details that you don't get with RZA, but RZA is really good, especially if
on the fly, you need
yeah,
some
on the
data.
fly, exactly.
Yeah.
Or if you're presenting something, or if you're talking to the community members, you want to give them some type of data that they can kind of gain an understanding of what's going on. So kind of continuing with that, this year on the boat, I did it in real time. So on a moving vessel, doing the RZA, very different because the specimen were live, so they were moving. The boat was also moving. So really had to develop a technique to look under the microscope. But
bring them back. Yeah, exactly. Try to tag them, bring them back. Yeah.
Exactly. But in addition to RZA, I was lucky to do other zooplankton kind of collections things. So a big one was identifications for some species, and we would collect those individually, and then that's what we send for kind of stable isotope analysis, so carbon and nitrogen concentrations for them. And collection started very early, 7 a.m. We use two types of nets, so a multi-net or a hydrobios multi-net. So it's essentially a big kind of cube apparatus, and attached is about 5 nets at about 150 micrometer mesh. And this would drop vertically up about 5 meters off the bottom, and then we'd kind of drag it up. And then the multi-net samples would be kind of put and preserved in formalin or formaldehyde, which is what our kind of main preservation for zooplankton was. And those would be sent for taxonomy. So the taxonomic assessments would be from the multi-net. And then we'd also do our what's called a bongo net. So there's 4 nets in total, 2 500 micrometer mesh sizes, and then 2 150. Those are our baby bongos, is what we call them. Those would be dragged horizontally in the water column for about 20 minutes. And then we do RZA assessment on that, as well as picking out certain species, kind of key forage species
Mhm.
for stable isotope analysis or
Mhm.
fatty acid analysis. And so that's kind of what my day to day looked like on the boat.
Oh my goodness, that
It,
is so awesome.
it was
That's
fun.
so cool.
Mhm. I did work at first, being a student and being not having experience in the field. I did work closely with kind of a supervisor, and we do it
Mhm.
together. But eventually, I felt I kind of gained the confidence over time to do the assessments on my own, which was very exciting. So,
Oh, that's fantastic. I
mhm.
absolutely love that. Thank you for walking through it. And thank you for doing such a good description and visualization of being like, okay, what do the baby bongos look like? What
Yeah,
does a taxonomic study and what does that actually look like when you go to get that data and to collect it? I absolutely love the walkthrough too of the importance of zooplankton, of why are we interested in it and
yeah,
what does this kind of like keystone species actually show us and why is it so important for the rest of the food web. I think that's fantastic. I
mhm,
love also the call out of the importance of studying methodology because I feel like that is how
yeah,
science works. And I think maybe if you're outside of the field, you might not understand to be a large amount of studies. And some of the most important studies are basically just checks of methodology or checks of new methodology
yeah,
versus past uses or past kind of ways that we do things. And I love that you do need both at the end, but the RZA gives you this beautiful snapshot that you can then use that data for the rest of the work going on the boat. Because again, all of
exactly.
the research is also all interconnected. This ecosystem's interconnected, but so is the research that's actually happening. I just want to take a little bit of a detour, something a little bit
Yeah, for
different.
sure.
And it's more just about the future and your future and kind of also the future of ocean conservation. But
Mhm,
I know something that you're really interested in is going to grad school.
yeah.
And I was wondering if it's going to be also focused on maybe methodology and zooplankton, or you're going to go back to being like, you know what, I'm going to revisit the sharks and I'm going to look at something a little bit different and do a 180. What is your kind of area of interest and what are you most excited about for a potential topic for grad school?
Yeah, so I'm kind of working on my application currently to
Amazing.
continue with the University of Manitoba, and it also in part kind of co-supervision with the Department of Fisheries and Oceans. So I'll
Lovely.
actually be staying with the same group that I've been working with for the past year, and I will be focusing on the little guys. We're gonna do my thesis in zooplankton, specifically in a kind of subcategory of zooplankton called meroplankton,
Okay.
and these are any sort of benthic inverts that have a pelagic life stage. So the
Cool.
pelagic life stage can be a couple hours to a couple months. It varies even within species or across. It's very kind of interesting, but our knowledge on meroplankton and the Canadian Beaufort Sea, which is where we study, is very minimal. This is a lot due to meroplankton look very similar morphologically, so it can be very hard to identify even taxonomically. A lot of the
Okay.
times when our taxonomy comes back, they're just kind of classified to their like genus or some,
Yeah.
not even their genus, their family or
Oh, wow.
yeah.
Okay.
So
So
it's
very high
very
up. Yeah.
very high up. It's very broad. So we'll kind of be focusing on that. We're gonna look at the taxonomy, but I'll also be learning or we'll be doing DNA,
Oh, cool.
using DNA to kind of identify species. So that's an up and coming methodology for zooplankton and meroplankton identification, which involves kind of getting DNA, running the kind of PCR on the DNA. And we're doing two types. We're going to try to do two methods. We're going to do bulk DNA from the sample itself, and we're going to try to use an environmental DNA method, which is
Oh, I've
essentially
never
from,
heard of that. I feel like that's very new.
yeah, that's still, yeah, very new. It's still very detailed process that's still going on, especially for zooplankton and meroplankton, but using water samples and then filtering those out to get eDNA and then running that. to
Oh.
determine which species may reside in the water column over there. And it involves a lot of very prone to contamination. So
Hmm.
it's very important that we kind of establish which steps we're doing, and we're working a lot with geneticists to kind of help us with that. So we want to see if we're able to use DNA and eDNA to identify species across the Canadian Beaufort Sea to kind of gain an idea of marrow plankton community structure in the area. In addition to that, we'll be kind of comparing the marrow plankton to oceanographic conditions using a multivariate analysis approach. So, in comparisons to salinity, temperature, chlorophyll A concentrations, all that fun stuff. So, I'm in for
All
for a
right.
very
This
long
is,
journey.
yes, exactly. This is going to be a fantastic PhD and also postdoc and probably continue. Like
We'll
the
see.
amount of work that you just described, that's fantastic in terms of research. That's very, very cool. I have a couple of questions on marrow plankton specific.
Yeah, for sure.
In terms of role, do they occupy kind of the same niche space as a zooplankton, or do they occupy the same role within the food web as a zooplankton?
They do occupy the same niche kind of pelagically. That's kind of also what we're trying to figure out is where specifically they may reside in the water
Hmm.
column in the Canadian Beaufort Sea, because knowledge, again, super limited
Yeah,
in that area. We don't know the specifics, but
their zonation. Yeah.
yeah, but they do. Not all benthic species have a pelagic stage. It's
Oh, okay.
dependent on even within species, or it's dependent, but that's our main thing that we're trying to figure out is the community structure and within the Beaufort in the water column. But typically, they do occupy the same kind of niche as the zooplankton. They rely on the water column to move, so that's
Oh, that's a
kind of what they
cool
do.
study. Oh my
Yeah,
goodness. That's also going to be such a cool study to look at changes in water column, maybe with time or past data if you do have it, to then
yeah,
kind of understand their role and where they actually are within depth
yeah.
throughout time and seasonality. I mean, there's so many ways that you could take this, but I,
Oh,
I absolutely
definitely.
I love it. I love that also that use of eDNA and DNA. And again, going back to the importance of methodology, I could see the eDNA, yes, prone to contamination, but it's also such a wonderful passive measurement that you don't have to do maybe more invasive ways of methodology with your actual specimens. If you can just get a water sample, that's
Yeah,
fantastic.
yeah. I didn't know that I'm not super familiar in like the
Mm.
DNA and genetics, so I'm very excited to introduce myself into that during my master's research. I think that would be a really cool uh, turn to get into. And metabarcoding is it's a big up and coming thing for species identifications, just because there's been kind of some studies going on about if it's able to identify more than taxonomy, or if it's for certain species, if it's able to identify more or certain groups. So I'm looking forward to that. Yeah, the eDNA is yeah, big thing that's going on with the just yeah taking a water sample. It's maybe less invasive, but I do still think the DNA will be a good kind of
Mhm.
indicator as
You
well.
need to have both,
Oh yeah,
especially with like a new methodology that's coming on. You need to have a little bit, you need to have a constant. You need to have something that's been peer reviewed for the last 50 years to be like, okay,
yeah,
yes, that data means this. But that's fantastic that not only it's new methodology, but a species that we might not understand, a species that we might not really be able to identify, and then understanding its ecosystem involvement and its place within a very, very important place in the Canadian environment and in the Canadian Arctic. So I think that's fantastic.
hmm
Mia, when I know something you're passionate about, I think it's come out in spades throughout this interview is ocean conservation and ecology and environmental health of the Arctic specifically is what we've been talking about. But I know that ocean conservation is something that a lot of people are going to be really, really interested in and they're going to want to support no matter what. But sometimes I think that that can get lost in science communication from maybe the scientific community to the public. Is there anything that you think the public should better understand in terms of ecosystem health and protecting the oceans? And then for support, I think we have the public behind us of being like, yes, we should support the conservation of the oceans, what should they support or how should they support? So maybe a misconception or something that they should better understand. And then how and what should the public really focus on in terms of supporting ocean conservation?
Yeah, for sure. I think going back when realizing how important annual monitoring is,
Hmm.
especially in the Canadian Arctic and even the Antarctic, is changes in ocean like temperature or ecosystems is not an overnight thing. It's a long term, slow process. And I think a lot of people don't realize that, especially with climate change. I know there's a lot of people who are still skeptical about whether or not it's a real thing or what are the implications of it, but it is a real thing. It's happening.
It's happening now. It's been happening for a long
It's
time.
been
It's very
happening.
real. Yeah.
So I think it's important that people kind of educate and understand that it's a long term thing that is going to take time. And I think just people educating themselves around what could happen, even to yourself, like seeing extreme weather pattern changes
Mm,
or seeing this like mercury in the Arctic, you
mhm,
don't realize that it is a self thing as well. So getting involved, donations. I know that science relies a lot on funding. Sometimes people don't get that funding that they need, and they're doing very important work around ocean conservation and studying and developing these methodologies. You know, reading up on that and kind of doing a almost self-reflection of like,
mm.
okay, what am I doing? Maybe if I, compost, if I do this, if I do less of this, that you can kind of determine, a little goes a long way in the end. If everybody kind of works on themselves, it can hopefully help the long term for the environment. But yeah, I think patience is a really big thing.
Patience and then patience and education. It sounds like on a follow up of the education sector, do you have any favorites that you go to be like, okay, what's happening in the Arctic? What's happening in the Antarctic? Is there any resources that you like to go to be like, this gives me a better understanding of research right now and maybe also the problems right now?
Yeah, for sure. Honestly, because we're in such a media driven kind of generation right now,
Hmm.
even Instagram, like following any type of account has articles. National Geographic is a really good one. There's a lot of like ocean Instagram accounts that post these publications, or that you can see, like even TikTok
Hmm.
has loads and loads of things that they're showcasing these animals. I personally am a documentary person, so I watch a
Me
lot
too.
of Our Planet. But yeah, I think the media is really important right now, and you can easily read about it or get educated on these social media platforms. And I think it's a really big and important thing to promote that on social media as well.
Oh, that's fantastic. Thank you. Thank you for
Yeah, for sure.
giving people some maybe some first steps because I think that's always maybe the first once you get in a rabbit hole, at least myself, I find out that's so easy to keep going. But that first
Yeah,
step of being like, okay, well, where do you go for ocean conservation or who to listen to? Because I think sometimes with the media, the amount of media that we have, it's almost overwhelming. So to be like,
yeah,
who is the source? Like National Geographic, the one that comes to mind is NOAA is a big one
yeah. No, for
or the
sure.
WHOI is also a huge one.
Mhm.
I don't know any influencers. Do you have any favorite ocean conservation influencers?
Not that I know of. I follow accounts literally like Ocean is a really big one. They kind of show lots of different types of marine animals and what's kind
Hmm.
of going on there. I follow quite a few shark accounts. They bring a lot of awareness to shark conservation, especially around shark finning, and shark fin soup is a really big kind of thing going on. But it comes up a lot for me on my
Yeah, they
it,
know your algorithm. Your For
yeah,
You page
my
is nothing but
algorithm.
ocean.
Yeah, but I think especially because, some of these science articles and publications can be quite heavy
Mhm.
and overwhelming for a lot of people, especially who don't have a science background or a biology background. I know even trying to explain things to my parents, like, I don't understand. So I think, yeah, even just those accounts that really kind of keep it simple and get you interested is a big thing. And Our Planet, again, super great.
Shout out Our Planet. Please go watch the documentary.
Literally.
Yeah. Shout out Our Planet all day. No, thank you very much. And thank you for kind of giving some easy to go resources, because I agree with you that I think academic papers, even if you're within the industry or you're within that study, they're like, woof, they can be heavy. Like even my own paper, I'm like, I can't read that like 2 years later. I'm like, no, it's too much. So I think it's always really nice to have a simple place to go to get information,
For
and
sure.
to have that building block. Mia, I know that we're at the top of the hour,
Yeah.
and I don't want to take more time out of your day,
Oh,
but
all good.
the last one is, I know something that you're passionate about is really just having conversations and being authentic in terms of the journey of science and being a student and starting to learn something that's really, really difficult and very complex. And then also moving from learning to becoming a scientist professionally and having your first kind of jobs in biology. Any advice there for other young people starting their career, or maybe in a position that's very similar? They're at school, they're in their third, fourth year. It's hard. They don't really know what to do, or they're freaking out. Any advice to other young people listening?
I think a big one is just to try and get involved as much as you can. I know, especially coming from like having no experience in science, a lot of these jobs are like you need 5 years, and you're like, I
Mhm.
don't. I'm just a
I don't
student.
have, I'm
I don't
22.
have that.
Yeah.
Exactly. So again, if any of the schools that you're at have a co-op program, or I know professors are always looking for help, and
Mm,
it's a really good way to kind of get involved doing kind of volunteering in a lab or any sort of volunteer experience. Even experience is experience, and I think when you do that, it's good to get involved, ask questions, show interest, because I think that's a big thing. If you're going and you're positive and you don't really know what you're doing, you'll learn, and you do that by asking questions and showing that you want to learn and you want to get involved. Another thing is to keep an open mind because where I started, like where I wanted to go with sharks and kind of shark
mhm.
conservation, is not where I had ended up, and I didn't think I would be doing and working with zooplankton, but it's something that's really become a passion of mine and something that I really enjoy working with, especially in the Arctic. So I think to, don't just kind of have your mind set on one thing. You might find something else that you completely didn't think to even look into. So that's a really big one for me.
I love that. I like the practical and also the mindset advice.
Yeah.
I like the practical of talking to teachers, because I think that is how you get a job. And I love that saying that you quickly just said casually, of any experience is experience, or a job is a job, basically. But you have no idea where it's going to take you when you have no idea the next step.
Yeah.
But I also love the idea of being open to learning on your journey, and then from that deciding. Thank you so much, Mia, for just having this conversation, talking about the importance of the Arctic and the Arctic ecosystems, the importance of zooplankton. And I think giving zooplankton a moment, because I don't think it really has many moments. It doesn't get the glory it deserves.
Yeah, we got to look out for the little guys. I'm telling you.
Yes, you do. It starts with the little guys, right? Like that's the most important part of the ecosystem. So
For sure.
thank you for walking through it and walking through your work up north and the importance of annual surveying. It was a fantastic episode, and just thank you for coming and having this conversation.
Yeah, thank you so much for having me. I had a really good time, and you're a
Oh.
great host.
Well, that's very kind. That's very, very nice. Well, thank you, thank you. Thank you for coming on Smoko. And for those listening, this is the Smoko podcast. We will see you next Wednesday with a new episode. So ta-ta for now.